Gene information is processed differently in prokaryotes and eukaryotes. Because prokaryotes lack a nuclear envelope, messenger RNA, mRNA, can associate with ribosomes in the cytoplasm as the mRNA is being formed. Translation of mRNA into protein begins before transcription is complete. Individual bacterial mRNA molecules often contain transcripts of several genes. By placing genes with related functions on the same mRNA, bacteria coordinate the synthesis of these proteins. These clustered genes are referred to as an operon. Because eukaryotic cells possess a nucleus, their mRNA must be completely formed and must pass across the nuclear envelope before translation. Eukaryotic mRNAs are modified before they are translated. Introns are removed and the remaining exons spliced together. A 5' cap and a 3' poly A tail are added. The processed mRNA travels to the cytoplasm where translation occurs. In contrast to prokaryotic mRNA, eukaryotic mRNA usually specifies only a single protein. In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA-carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. There is also a third site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of one codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of one codon. As the shift occurs, the two amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.